should show that it's recording, but it should show as a red dot somewhere, but I don't see it. Okay, now we
So
see
it
it.
has
Okay,
started.
okay, good. All right. So, so what we are doing is we are looking at life on Earth and how it originated, and this is a run up to what we had done earlier, which is to understand how systems biology really grew into this field. And one of the questions we were then trying to answer is, how did this complexity that we know as life really became existent on this planet, right? And and it is a difficult question to answer. And so we will break this lecture up into 2, 3 parts. And the first part will be to look at how these animate objects can get self-organized into something that we can intuitively say, if it made that jump over the gap between animate to inanimate, it would give you a living cell. So that gap still exists, and we don't have an answer to that. And it will continue in this fashion over a period of time until scientists break through this barrier. Okay. So if you look at this figure, which is given to you on the right side here, it shows you on the left hand side the frequency of occurrence, and on the right, on the y and the x-axis, you see complexity. And as you see that in the earlier part of the evolution on planet Earth, we'll find that that you had all these microorganisms that were living, and they essentially formed a, a kind of a skewed distribution in terms of complexity. And then we have what is called the minimal complexity that is required for the growth of a cell, and we do understand that that is needed because as we discussed before, that only when we have a complex enough system do you see what are called emergent properties, and emergent properties are necessary to define what life is. And as we go forward along the x-axis on the bottom panel, we'll find that the bacteria over a period of time, as it came to the present, evolved through, as we know, all the various biological species, and finally give us the most complex species, like which are called, well, we are the humans, who are the final uh, intelligent species on this planet Earth, right? So, and now we are questioning, how did all this happen? So that itself is a huge miracle, or a huge jump in terms of consciousness. So all we know is that, that this life had started on this planet over 3.5 billion years ago. And we'll see the next slide that, if it was supposed to happen, then, why on planet Earth? So, so this is, a question that many people ask, and those who are in this field who try to understand these aspects of the evolution of life, essentially look at this question. Excuse me. And so, um, the other question that people ask is there a silicon-based life somewhere? Do we know if carbon-based is the most favorable life, and that is what we'll expect even in other forms of life across the universe? We don't know, right? So, so all we know right now is that because carbon has such a rich chemistry, and the chemistry of concatenation, that means one atom to the other, and the hybridization that takes place, the sp3-1, sp2, sp3 hybridization, which is there, and along with it, the rich chemistry of phosphorus, nitrogen, and hydrogen and oxygen together combined, which gives you what life is on planet Earth, right? Now, the question whether or not
Sir,
life, yeah, yeah.
in case of we are considering that if there is a silicon based a life on some extraterrestrial planet or in some other galaxy, so in that case what will we be considering as life, like what properties we are considering to have there?
Okay, there are certain, I mean, I don't remember all the laws that are there, but there is a group of these laws which are there, which essentially will tell you that if it satisfies these things, which are abstract, which are the laws that we can assign to living organisms, then that would be translated to the similar thing in silicon life. So, for example, the one of the things is that living organisms can create other living organisms. Living organisms
Yes, sir.
uh, um would require energy, would require resources, and based on that, it will create its own requirements that it requires in terms of molecules of it that are required to build up its cells, and so on. So there are these 7 or 8 laws, I think, that have to be fulfilled, and what fulfills for a system that we have for biological systems also has to fulfill for the silicon life. Just give me a second. I'll just go and get a glass of water. I thought both the video and the thing were off. All right, I'm back. So whatever has between this gap, you can ignore. All right, so I'm going to play this left-hand side video only. The right-hand side I won't play. That's a much longer video, and I think you can look at it at leisure. So the reason I'm going to play this, and maybe I won't play it. I'll just tell you what it is saying as it goes through, because I don't think you'll be able to see you'll be able to hear the voice. So it essentially describes what is the anthropic principle. And
imagine entering a room.
Can you hear this?
150 dice on a table.
Yes, sir.
Yes, sir.
Oh, if you can hear it, then it's
you
fine.
immediately assume that something is wrong with those dice. They've not been thrown at random because it's very improbable they will land just like that. The same puzzle is being faced by cosmologists as they try to figure out why is it that certain constants of nature, among them the cosmological constant lambda, take on the value they do in our universe. The value of lambda is very, very small, and it's thought to be responsible for the accelerated expansion of the universe. However, we don't understand why it is that it is so small and yet non-zero. There is more. If we change the small value of lambda by just a little bit, in other words, if we change the outcome of one of the dice by just a little bit, the universe as we know it would not exist. It would be a very different universe in which we, as human beings, would not be able to thrive. So why is it that we live in such an improbably finely tuned universe? One possible answer comes from string theory, which postulates that we're actually part of a much grander reality called the multiverse, in which different universes coexist, up to 10 to the 500 of them, all of them with different values of the cosmological constant. And so only those universes which have the value of the cosmological constant that's just right for us will be the ones in which we, as human beings, can possibly live. And therefore, all of the other universes are impossibilities, and we are left to live in the one improbably fine-tuned universe that we observe. That is the anthropic principle.
Okay, so you get the idea, right? What the anthropic principle is. So
Yes, sir.
yeah, so this is a very nice. There are several other very nice videos which are there, and one of them is from Stephen Hawkins also. So if you do a Google search, you'll get several of these which are discussing how life on Earth is a possibility, is one of the options that happens out of string theory and the anthropic principle. All right. So this is just an overview. What is the scale of evolution? And we know that the Big Bang happened about 15 billion years ago, and that our solar system was formed around 4.5 billion. And the first molecules that started interacting was around 3 to 3.5 billion years ago. And then it took over another 2.5 billion years for the first cells to start appearing. And then it took another billion years for the evolution to take place to fish to reptiles to mammals. So, and it's only in the recent 1 million years that this whole thing started speeding up, and that's what took about 1 million years to develop from mammals to us. So that's a brief history of the evolution of life. So you can see that this evolution that has taken place is actually something that that we can only appreciate in terms of the huge scale of time, right? So the time scale is tremendous, and it does come as a real surprise to us that all the knowledge that we know and have accumulated really comes from the last 500 years, maybe at the most, in terms of the scientific knowledge that we consider as modern science, the way we have been developing. So that is really a surprise. All right. So, but life as we know it was created from chaos. And there is just no doubt about this. If you just look at the primordial Earth, then the primordial Earth was very, very complex. It was something that was, the atmosphere was, being pounded by meteorites. The atmosphere was inhabitable and so on and so forth. And I will not go into all of those things that have happened. And that is also, there is a classification of the conditions on planet Earth and how it changed and how that gave a rise to life as we know it. But leave that aside. So if you leave that aside, then what we find is that there are three things that come out of this. Where we talk about self-organization, we talk about complexity, and we talk about emergence of new properties. So what it means is that if the molecules that are there, these molecules that have been used to build life or were participating in the formation of the first cells, if they did not self-organize, it was not going to be possible because the environment itself was chaotic. Then the other thing is that it had to be complex enough, because if it was not complex enough, then it would not have had the opportunity to bring in this vast diversity of biological world as we see it today. And obviously, it is from the first two that we can have or expect new emergent properties coming up, which are much more than the fundamental properties of the individual components that build up to this cell. So this is a very fundamental problem of biology, and it concerns with various kinds of origins and forms and functions that are associated with biology. And we really don't know much more than this at this stage, except we can only conjecture, because quite a bit of the information that is supposed to have been clues are lost because it's so old. We don't have it here anymore. The rock formations are no longer existent. So one thing is for sure, that the second law of thermodynamics, which says that entropy, which is disorder, is continually increasing over time. It should be third law of thermodynamics. This is the third law of thermodynamics. It always increases with time, and therefore the decrease of entropy in a cell is only permitted if there is disorder in the environment. That means entropy in the environment increases, right? So that said, let us look at what was the first time that somebody in this modern era thought about biology as the way we know it in modern biological sense. And you would be surprised that this comes from a philosopher by the name of Immanuel Kant, a German philosopher who talked about dynamical systems theory, and that's the exact word that was used, dynamical systems theory. And he was a proto-theorist of complex dynamic systems. He talked about the nature inherently contains not only automatically mechanized processes, but also natural, creative, self-organizing thermodynamical processes. And also said that nature essentially grows and has a complex dynamical history. Now, these three tenets which are being stated here have come after a lot of intuition, introspection, meditation, as they put it, for these philosophers. They used to write meditations on fire, something like that, and then discuss the properties of fire. So So he was one of the well-known, and even today is very well-studied philosopher. And he talked about biological systems as opposed to other physical systems, and how the nature of biological systems could or should be inherently different from that of physical systems. And this term, which is such a modern term, dynamical systems theory, was first found in his notes. So he was a great philosopher, and there is a time when it's said that he wrote something which was criticized by the general philosophers of his time, and that made him become very introverted, and he kind of shut himself up for about 10 years. And after he came up, he had these huge body of sign of work that he had carried out in those 10 years, that is such a huge boon to now both to philosophy as well as to science, because he questions the very fundamentals of how does matter form, why should matter be there at all, things like that. So these questions as fundamental as those are being answered, or at least debated and uh, discussed in substantial detail by Immanuel Kant. So if you can, I have a note of his philosophical, so I'll have to unshare and then share. So let me unshare this for the moment. And what I'll do is I'll share another document which I kept here so that you can see that. And let me see if I can share this screen with you now. I'm just doing this. I hope you can see can you see this screen with the Adobe?
Yes, sir.
Okay,
Yes, sir.
and yeah, then you see here, this is taken from a writing which discusses his work, but these are in quotes. So you can see the general mathematical theory of complex dynamic systems called dynamic system theory, and this mathematics of DST was richer than the PRA. So it actually goes up. So this is all, otherwise put Kant's self-organizing system as are all living systems. See, so he talks about self-organization at this stage. So he is talking about, the notion of self-organizing used to contemporary theorists of self-organizing thermodynamic system is broader than Kant, in which he includes non-living complex systems as well. And the rolling hexagonal Bernard cells that appear in water ahead of and thunderstorms. Kant's self-organizing systems are all holistically, causally integrated or autopoietic, such that the whole and the parts of the whole and the parts mutually produce each other. So there is a whole, and he talks about ontologically emergent, causally efficacious properties. So these are little things that has been taken out from his work, and he talks about natural entropy. He talks about natural generative teleology. And so I've marked a little bit here, and this is for your interest. There is therefore for Kant an irreducible explanatory gap between biology and classical or Newtonian physics, which is the same as the contemporary explanatory gap between the nonlinear, non-equilibrium, non-mechanistic dynamics of self-organizing living organisms, or rather living organismic thermodynamic systems on one hand, and the classical linear equilibrium mechanistic dynamics of inertial non-living physical system on the other hand. So it's very wordy, and you can see that he uses a very complex adverb and ad phrase to discuss these things. So I'll go back to the presentation. So are you able to appreciate this? Yes?
Yes, sir.
Yes, sir.
Yeah. So you see, I just took it out from something that was a kind of a treatise on his work written by another philosopher of modern times. But you can see how the words that were highlighted or words that were in quotes come from his own work, and how modern it sounds, right?
Yes, sir.
Yeah. So anyway, I'll include this and one or two of his works in the just for reading, general reading. So you see how it feels. So what I'm going to do now, the next part of
His works are in English or in German?
Originally it was in German. These are all translations. I think I took it from a Harvard philosophical site. So I was reading and I found a link to that, and then I found that they have a huge repository of discussions on his work. So you can check that out also. So, but we don't have time. No, all of us, what happens is, and actually, I think you will all agree that this pandemic has made us realize that our method of education has to change. So,
Yes, sir.
I mean, and you may not be knowing it, but the previous cycles of students that I had, I've been discussing this for 3-4 years actually. That this before the pandemic, I've been discussing with students that this is not really the way we should be teaching students, because there is so much of information there that we should not be going and just repeating that information for the students. Let them find it out, and in the meantime, we discuss mainly the ideas that can lead them to think on their own. So that is the way I would like to do it. But I don't know. We'll see if I can. I'll try to think about it. Let me get well from this COVID illness, and then I'll do maybe something about it this round. So in the next part of my talk, what I'm going to do, or for this lecture, is so we have established that what we had said earlier, that systems biology is a description of biological systems, and that was necessary because biological systems are so complex that the older methods were becoming useless to describe it, and so a newer method and more complex methods, more technically sound and more advanced methods had to be used. So that part, and that we now proved in what we said in this first part of this lecture, that yes, what we said about life, as we understand in biological systems, is really true, and this complexity was a necessity, and that life had to originate because of anthropic principles. So we have kind of established all of that. Now what I'm saying is, the next part of it is that I'll lead you to the gap, and then we'll jump the gap and say, well, that's where the cell happens. Okay. So to lead you to the gap of where all this organized components of inanimate objects, if I may call them objects, will actually then give you a component that looks like a cell and would have otherwise inanimate properties that a cell would possess. And then if we make that leap of faith, then we get the cell. So I'm going to break this part up into 3 groups of study. One is self-organization of physical systems.
Just sharing the screen. Yeah. The screen.
Yeah, what happened?
I guess your screen isn't shared yet.
Screen is not shared? Oh, what happened?
No, sir, your screen is not shared.
I'm so sorry. So I'm doing that. I thought I'd shared it. Then I was talking away. Now it's okay?
Yes, sir, now it's okay.
Okay. All right. So what I'm going to do now is break this next part of this lecture into 3 components. The first part to show the physical self-organization, the second part to show the chemical self-organization, and the third part to show the biological organization, and how one leads into the other. Okay. So, and all these are by examples. So, and you know that these are the very well studied experiment, where actually you have a very flat plate, and on this flat plate you have a uniform temperature. So that is something very critical, that how do you maintain the uniform temperature across the flat plate. Now you allow this flat plate temperature to rise gradually, and what you'll find out is that from a random movement of particles, which we all know will happen because of the Bernoulli's movement that is there because of the random movement, right? But then what will happen is, at some critical temperature, a bifurcation in, as we know it in terms of system definitions, will occur. But what does it mean in terms of the physical system? That convective currents suddenly appear. And whether or not the convective currents will be, let's say, in this direction here or in the opposite direction there, that you cannot predict. But it will happen. And this is one level of organization that you see just by changing of one parameter gradually and over a range until you cross what is called the critical temperature. So you see that just by doing this, you are able to organize what was essentially random molecular motion. And it's very easy to observe because if you just put a drop of some dye, you'll find that the dye will move in a certain direction. And in other cases, that dye will be moving just random diffusion. So these things are very well studied, and this what we call as these Bernard convective currents, which are there, are very well known. Okay. The next one is chemical self-organization, and this was also studied in the 1970s and very well published. Belousov-Zhabotinsky reaction. So this reaction, there are two components. You have you have bromate ferroin and bromomalonic acid system. So this is the bromoferroin, and that is the bromomalonic acid. And this first part acts like a catalyst, and the second part acts like a reductant. So you have two opposing effects. One is trying to activate and get you the reaction. The other one is trying to reduce and contain the reaction. And the self-oscillation takes place over a period of time T. And there are two phases that are distinctly seen here. The T1 phase is the phase where the Fe3 concentration increases, and the T2 phase is the phase where the Fe3 concentration drops. And I'll show you this video now, and you can see this thing happening here itself. So you can see the spot at which this is going to come up, the next green spot, you cannot predict. So of course, in this one, if you see the second time, you can predict it by prior knowledge. But you see how it happens, how these patterns start forming. So this is also an organization out of something that is not organized, right? So it's a completely uniform movement of particles which will be there, giving you the orange color. And then you start seeing the organization of these reactions take place and the conversion to ferric ion. And those are going to move out in green circles, as you can see them. Clear? So you see, this is another self-organization. And in fact, this idea of these circles in a plane where you have closed loops on a plane, if this was translated into what we talk about in control theory, if we looked at the phase plane analysis and then we see these circles, then essentially this contains information, and that information is contained within the circle. So often memory systems that are there, that people work with AI, ML, and other things, they are also have these properties where you have a circular trajectory if you were to plot it on a phase plane. Circular means not circular like this. It means it has to be a closed loop, right? All right. So this is the original article from which which people are, and the other one, of course, is an experiment that you can do in a lab. So this was the original article where they showed that this could happen, and the wave propagation is approximately 0.01 centimeter per second, and he's given the conditions. The paper gives the conditions under which this happens. And you can see that, given a very specific condition, sodium bromate 0.23 molar, BMA 0.16 molar, ferroin 0.003 molar, and sulfuric acid 0.26 molar, and temperature 14 degrees centigrade, you'll get exactly this, and you are able to reproduce it. And the photographs have been taken in this one because we didn't have a video in those days. They took the frames every 30-second interval. And you can see how this grows and gives you this pattern. Yeah. All right. So that was the second part. Now the third part. So the third part that we are going to talk about is how biological orders took place, right? And so if you, all of you, has someone among you studied the origin of the genetic code or the origin of these molecules in a standard book like Leninger or Stryer? Yes.
So we have looked at some parts as a part of biochemistry from
Yeah.
Stryer.
Yeah. So this is, I think, this some part of it is there where they talk about, the first synthesis of the amino acid. So does anybody know how much energy is required from one peptide bond? I've given the value here in 1,000 times more, but what is the value, actual value? It's 2 to 4 kilocalories per mole. And at 25 degrees centigrade, if you just leave two amino acids side by side like this, it'll take some hundreds of years for them to form the peptide bond. Okay. Now, given a billion of years, that seems quite fine, right? That you're looking at really something that's happening very fast in the scale of a billion years. But really, you will not see the complexity that has happened just allowing that to happen. Also, I mean, within those 100, and we know how things happen at this, when we look at molecules. So the scale at which things happen at this scale is, the vibration of the molecule is very fast. So hundreds of years would mean things would have changed between two amino acids sitting together next to each other, would not have been isolated from the environment. So there'd be many other things happening in the environment. So that's not possible. So what is the question here? The question is, when did the first biological molecules form? And of course, as I said before, answer is not known. But what we do know is what could have led to it. And we can conjecture as far as possible, that we can't go beyond that. So what we can say is that, we know, for example, that amino acids cannot polymerize because of the dipole-dipole interactions. We also know that if we looked at the minerals that are present, for example, kaolinite, then it's possible that they could get adsorbed and form a aqueous solvent and in the solid solution, and then that could have kind of helped, because it would be now 1,000 times more we've 1,000 times faster. I don't know, about 1,000 times more amino acids were. So what it means is that the rate would have gone 1,000 times more, because the vicinity at which they would be on when they were residing on the kaolinite would be close enough and for long enough duration that this forms. And there would have been exchange of other principles, maybe of ions from the kaolinite, which would help this transfer to take place. It's almost like a solid catalyst on which they were sitting. Preferential synthesis of pentoses and hexoses would have formed first. We know that. And also we know that this polymerization would have been something that would have also been, So we know that we had this polymerization of the preferential L amino acids, and perhaps the presence of something like kaolinite could explain that, because of the presence of such a protostructure on which the amino acids could sit, there would have been a enantiomorphic selection which would have taken place, and that would help only one kind of amino acid to combine with only one kind of other amino acid, so only the L type. So that's how things must have happened, but really we don't know. I mean, this is something that we know can happen in the lab, but how did it happen, we don't know. But the fact remains that yes, we did get peptide chains in the primordial soup that was there existing at the time of the first creation of life. Let's put it quote unquote. So the other thing is, obviously, the cells, if they were just out there in the environment, this will never lead to what we know is a cell with a cell membrane, unless we had something that separates from its environment. It's only then that things can happen more specifically, because otherwise the randomness is so huge that even a billion years would not have been sufficient to create what we know as life today as it exists here. So as you can see, that you could go back and look at the organization that takes place at the level of micelles and vesicles. If you were to take, an oil, a water, and mix it with some surfactant, you'll get what are called micelles, and they are very specific in terms of what concentrations of each you use, what variety of each you use, and what is the shape that it takes, right? So look at this vesicle here. It's like a bilayer here. So we have you can see that they're created in such a way that you have relative attraction on the so this would be there is a it just depends on what is on the outside. So if you have aqueous medium on the outside, you'll have to have a charge group on the outside with the hydrophobic group going inwards. Similarly, if you have aqueous medium inside, you'll have to have a charge on the inside and the hydrophobic group will be there, right? So you have a head-to-head repulsion that takes place, and as you can see, the long chains that are there, that is inside here, then that helps to form the lipid bilayer. Now, the micelle that we say has an optimal surface area per head group, that's very important, because you see how you're going to pack it will determine whether or not you are going to be, how you're going to pack it is dependent upon the shape, the geometrical shape of this molecule here. All right. So so there has to be some kind of a minimization of energy based on the surface per lipid molecule, which is participating in the bilayer. All right. Oops. Now, there is a very specific equation that actually governs how these vesicles actually will form, and that equation is given here. So V is the volume of this geometric shape that would enclose the molecule. A is the head surface area, or the surface area that is exposed like this. L is the length, the radial length from the center to the center here. And then you have this equation on the right hand side, and there are two parameters, H and K, and that H and K is given here. H is for a sphere, it's 1 by r, and K is 1 by r squared. So H and K defined as here. Very easy to see. For a sphere, R1 and R2 are equal, 2 by r, 2 cancel out, you have 1 by r. For a bilayer, it will be a planar infinite curvature. So 1 over infinity, 1 over infinity, 0. And 1 over infinity, that's 0 also. Just excuse me for a second, I'll just drink of water. Oops. So I'm back. All right. So this is something that tells you that depending on these parameters and the values that you see here, you could actually go around here and form anything, any shape that you wish. In fact, that is given in the next slide, which is shown here, that you could form any of these shapes, and the values of V versus delta is given here. Delta is the difference of the area, and V is the dimensional volume to area ratio, which we previously discussed. And you can see that you can get all of this. So now I'm going to show you a slide, and I'm going to ask you. So what do you think this is? Can anybody tell me? Image on the right hand side.
Cells.
What cells?
RBCs.
Yeah, let's see what it says here. So this is an AMF image of a polymer vesicle. The aerosol except arises from the evaporation of water from the vesicle interior, leaving the prominent rim. So you can see you can do this, and you'll conclude the same thing as I would have concluded also the first time I saw this. You're right. It looks like an RBC, right? Now you tell me which one of this is between these two, which is a real and which is an artificial. Anybody?
The left one looks artificial and right one perhaps.
Sorry.
Left one looks like artificial and right one perhaps it is cell membrane, I guess.
Okay. I think you're right. This one. So left one is a vesicle, right one is a diatom. And the genus block genus, the high genus number means that the higher the genus number you'll have, it associated with the hole that you see here. So the perforation that you see arise because of another factor which we have not discussed here. It's called the high genus number, and there's another parameter which is there, which if the value of that is high, then it gives you these porous structures. What about these two? Anybody? Next two? Yes. Okay. So I'll just again have to go out of this. Oops. Where am I going? Oops. I don't know what's happening here. So yeah. So this one is, sorry about that distraction that happened. So this is a 10 image showing you barium chromate, whereas this one shows you the ApoE amyloids. Can you see? So they're very different in nature, but very similar in structure. So those of you, so today is now I can still continue, but I don't know what the rest of the class would like to do. Would you like to discuss or continue with this? Yes.
We can continue.
Okay. So I don't know. This one is not asking me
I
for.
get a question like.
Huh?
Sir, like there is a equation that you shown.
Yeah,
so
yeah.
based on that, we can say that because we can explain like why E. coli cells divides into equal daughter cells and not like unequal. Yeah.
No, there are many. No, for that there are many other. There are many other parameters that dictate that. For example, how the actin molecules come together at the juncture and help it to divide. Many other factors are there. So let's not get there. But finally, if you look at the fundamental, yes, the shape that is going to be it's going to be initially like two bacilli will be there. So in that case, these are going to be cylindrical, right, for E. coli. So they'll form a kink somewhere and then they separate out. So that I don't know, but we can see that that there will be some differences that are going to occur at the juncture. That is defined by these equations also. So it's going back and forth. So if you're constructing a vesicle of that nature, then you need to know what parameters are there. And if you want to describe it, that's a different thing. So here all you're saying is, well, my equation is fine. But that doesn't really add to the value of understanding why the cell divides in the way it does. It only explains the physical phenomena that divides. So that's not very interesting the other way around. Okay, we'll continue. And you tell me when to stop, you guys. We'll continue like this. So I think we should, yeah, I think we can continue with this only. So what you see is that up to this point, we saw these three things that we talked about. So we talked about the physical, the chemical, and the biological organization in terms of the molecules that are required, and how all of that happens. So that we all accept. So what it has shown you is that, well, you can have a vesicle, and in that vesicle you can have these proteins, and you can have all of these other compounds which are there, and that could be encapsulated inside that. And by some reason, if they are combined together and they're all holding together in one unit, they could give you new properties because it's so complex. So now, whether or not it actually happens and how it happens, whether or not we can reproduce that today, we don't know. Right? We have not come to that level yet. So all we know is that that jump took place. That means if you can imagine that even today we can explain very precisely using a mathematical equation how a vesicle shape will be by adding different compounds and working out the parameters and doing it, then obviously that is also possible in the real life situation. That vesicles of different shapes can form, and once they are formed, they can actually enclose. If they are in a environment which already has these compounds, it will actually enclose these compounds. And if there is a high enough concentration, it will be complex enough that something happens. Now, where it happened and how it happened, and how that jump took place, so that it gave you the property of life, which is self-replication. One of the important properties is being able to regenerate itself. That gap is not yet known, and so there are of course many more missing links. That is the major missing link. So we see that from the physical to the chemical to the biochemical, we do see the self-organization take place, and the complexity increases. And as it increases, it gives you the first cell, and that one is called the last universal common ancestor, or it's called LUCA. Okay. So what you see here is chemical evolution. You have the common ancestral plane on which all the evolution starts from. So at least there would have been one such cell, because we know that life has to be created from cell. So we notice some important fundamentals here. Though it's putting the cart before the horse, that all life, biological life on planet Earth, is based on the DNA code, which means that the CTAG, right? And all the proteins that are there are based on the 20 amino acids that we have in the natural world. We have some exceptions here and there, but that's primarily this. And that the translation of information from genetic code to this takes place to the RNAs, where the T is replaced by the U. So you have UAGC instead of TAGC. All right. So you see here, then of course the whole component comes in. You talk about archaeobacteria, eubacteria, and eukaryote and eubacteria, and then the breakdown as we move forward, into higher animals. So all of that happened. So, and where I've taken this figure from, that reference is given here. So do we agree to this fact then, that yes, that it is possible? I'm always making a mistake in this direction. So do we agree to this, all of you? Hi, everyone.
So I believe at some stage it must have happened. So maybe it is possible, but we're just not there yet.
Yeah. But you agree at least to the run-up to that I've shown you, that this seems to be the case, right?
Yes, sir.
Okay. All right.
Sir, but there are other theories like life was from, like there are microorganisms which can even survive in a space. So how there may be?
You're right. There's a what's called, I forget the technical term, but essentially it essentially means that it could have come from extraterrestrial source is possible. That you could have had a meteorite which had cells that fell on Earth, and then that became the seed for all the cells that are here. That's also possible. But wherever it came from, there some principles would have been there which have been similar to this, right? So the fundamental principles are not going to be shifted, but source could have been shifted. Yes, true. Okay.
Yes, sir. Yes.
All right. So here what we see is we see that the self-organization that we talked about, and this particular slide that I'm picking up the photograph from, I mean this image from, I mean I had started teaching about Kant and all these things much before they published this in this paper. So Nature Reviews, I was quite pleasantly surprised that they actually also took Kant as the first source. So this actually comes after my initial teaching. So when I started teaching system biology, I used this slide on Kant, and then I found that, well, somebody has also used the same thing. So really that is the earliest reference that we have. And if you look at self-organization, which gives you cell polarity and principles of that being accepted, widely among the scientific community, that's around 2003. That's where a lot of work was also revisited in terms of what and how self-organization gives rise to vesicles and biological systems. And that's when, also this Craig Venter started his work. And so the self-organization of cells talked about this. The version that we are talking about here is, around 2005 to 2007, when, some work was published based on the work that on the likes of what Craig Venter was doing. So there's a huge zone here, but we are looking at the most we're looking at how many years? 21, so 300, 200, 300 years, no, not more than that. Okay. I'm going to show one of these videos, not show the other one. So this is again self-organization of magnetic dipoles. So see how other systems are there which exist where self-organization also can happen. So there are two poles which are there, and these are all magnets which are at random, as you can see. And just based on one force, how it separates out. So what does this really tell us now? What is this? What is this video hinting at? Anybody?
It is like whatever happens in biological system, it can like explain.
No, I don't know. I mean, so I'll just stop it here because the discussion that we'll have is going to be better than this. So I'll just stop the video. So I'll just see. So among some of our faculty colleagues, we had a discussion a while back. I mean, it's not now, maybe 6-7 years ago. So it's one of the very nice. It just happens sometimes. You just get into this flow and you have some good discussions. So I don't know, and so this person is a physicist. He's a quantum physicist, so he knows quite a lot about all those things. And just like you see here, so this separation, there was a force that was required, right? Right. So there's a force that is required, and so it appears to him, I mean, after his much study that he's done, and he's a very senior faculty, so and he's been also pondering about this, and you'll notice that quite a lot of these books that were written in the turn of the 19th century, right, 1900s, early 1900s, when we have this these physicists talking about life, right? What is life, for example, right? That book. So quite a lot of this discussion took place then, based on those books and other things we were having a discussion. So it seemed that at the fundamental level of matter, when we have these inanimate objects, which are these molecules which are organizing and having these specific properties that are there has to be another force coupling that takes place to give you what is then finally translating to life or living cell. And that coupling of that force is still not known. And obviously we are still in physics, we know that there are higher dimensions and other things. And we can only visualize up to the 3 dimensions, and that we know up to 4 dimensions that whatever we learned till this point exists. But really a huge contribution will have to be there for physics to understand what life is, both physics and chemistry, because where does that coupling take place is not really known very well yet. And it might take many, many years before people do understand that. So we can mimic it, but we can't really produce it. So that is something, food for thought for you. Think about it. So now talk about the genetic code and evolutionary theory. Okay. So so we do know that that there is a universal genetic code. All of us agree with that, and we know that that is what is DNA is made up of all. And then we also know that there is a certain optimality that is there, right? And we agree that this genetic code that we have is optimal. We also know that this genetic code must have been created before the first living cell came into being, because otherwise, how would you have this information being transferred from one cell to another? So very clear. Then we also know that there is a horizontal transfer, because if there is no horizontal gene transfer, then this rate of evolution would not have been as fast as it is. It would have been much longer. And we talked a little bit about this in a different context while we were doing the slides. And then also there's a probability of an error which is there built inside this translation of this genetic code to proteins. And that is something that actually has pushed evolution to take place. It's one of the factors that leads to evolution. And obviously we know that that there is a standard code, and it's called 1 in a million, because we could talk about a frequency. There's a fraction of random codes F that have a lower cost than the real code calculated. So I'll talk about this a little bit, and that is 10 to the power minus 6. So what people think is that the evolution must be some kind of cosmic principle for the universe and not just life on Earth. So if we look at all of these things, then something similar, if like we talked about silicon code, right, a silicon life, something similar should also be existing in those conditions also, right? So yeah. So let us just so I'll just talk about this thing, and then we'll stop here, because it's nearly my voice is getting a little itchy again. So let me just discuss this a little bit here. So the probability of an error is 1 in a million for the human genetic code. So the question goes back to whether this genetic code that we have right now is actually optimal. How many of you think this is optimal? How many think it's not?
So optimal as in?
Optimal means that could this genetic code be made better so that you would have lesser error? Could it be made better that you would have a wider range of amino acids that could be incorporated, things like that?
Sir, in terms of we can make, perhaps we can make, but like in nature, it is perhaps optimal. Like there are now scientists are making a new synthetic base pairs. So and those are like more efficient in terms of like replication and etc. I guess.
Okay. So we'll just conclude with 2-3 points I'll make now. Huh? So because it could get late otherwise also. So 2 points to keep in mind. So what Ratan is saying is something on the similar lines, but I'll just make those points a little more clear. There are 2 points to be noticed here. One point is that once the code is in place and the biological world is using it, that means you have the genetic code that gives you the proteins. it's going to be very difficult for the biological world to convert it again to make it further optimal. Because every time it makes a change, that change has to transmit to the entire biological world, right? So it evolved over a period of time. Now where it is, it is. So it gives you a reasonably good. So it gives you something which is reasonably good. That's fine. But beyond that, it does not. It cannot be made further optimal. But we can test it because now we are thinking about the genetic code. So how do you test it? Well, you say that if I made one change in the genetic code, what is the error that happens and what is the consequence of the error, and we put a penalty point for that error. And how we define the penalty point and other things, that is really a little complex, and we won't get into the discussion of that. But just assume that there is a way that you can assign a penalty point. It's just like an optimization problem. So if you have an objective function, then if it makes a wrong turn when you're making the parameters move in a certain direction, you give it a penalty point so that it doesn't go in that direction. Simply something like that. So and if you do that and then you create random codes, thousands and thousands of random codes, or thousands and thousands of random changes, then what you notice is that, just by making random changes, you don't necessarily improve the penalty. That means you reduce the cost. It doesn't always happen. There are very, very few changes that really give you a lower penalty, right? For example, I'll give you another one. I'll give you an example. Let's say there is an amino acid that has got 6 redundancy. That means there are 6 codes for it. Genetic code for it. If I made that 4 and assigned those 2 other codes to something else, would that improve the optimality of, or reduce the error cost that would be occurring in the biological, in the way we define the biological translation, let's say, in formation of from the gene to the protein? Can we define that, and how much would that cost be? So then you look at this, and then you look at the frequency of these things, and then that's how you define. So what you'll find is that there are very few things that can actually give you a higher or a better code in terms of lower cost assigned to the changes that take place. So with that, let's end this lecture. If you have any questions, please go ahead and ask. Yes, anybody? No questions? So shall we then close this lecture? Yeah?
No questions currently.
Okay, all right. So I'll stop the recording and conclude the lecture, okay?
Sure, sir. Thank you, sir.
Okay.